Okie dokie, today we're talking about chemiosmosis. This is going to be a quick one, and this is just the making of ATP by with a proton, or technically a proton is just a hydrogen ion gradient. And so how does this work? I'm going to talk about this in regards to photosynthesis and the light-dependent reactions, but kind of just what we've got going on here. And I have a picture above, but I'm going to draw it. So we have our cell membrane, and it has in the light-dependent reactions, we have photosystem 1 or photosystem 2, and we have photosystem 1, and we have the ECT, electron or ETC, the electron transport chain. So blah blah blah, electron comes through, it pumps the hydrogen ion in, right? But what is that? And so now we have a high hydrogen ion concentration in here. Cool. But now that we have this high hydrogen ion concentration, what are we going to do with it? And that's basically where chemiosmosis comes in. And this is just using the ATP synthase. So it has a shape and then a shape like that. And so it's an enzyme that the hydrogen ions or the protons are able to flow out of, and they're moving through the enzyme is what allows ATP to be made. So ATP synthase is going to take ADP and an inorganic phosphate and bind them together to make ATP, excuse me, as the hydrogen ion moves back out and diffuses out of the cell. So that's really all chemiosmosis is that as the hydrogen ions, they're pumped in by the electron moving, and then they flow out through ATP synthase to make ATP, hence the name ATP synthase to make. And A is for the protein, because A is for enzyme, and enzymes are proteins. And that's chemiosmosis. Thanks for watching.